Good evening, everyone. I hope you can hear me loud and clear, and we are back with yet another event. I am Caroline, your host, and I'm very, very happy again to be here. And thank you already, everyone, for joining us tonight. We do have a very interesting topic to discuss. And as you can see, we have another special guest. This is Dr. Valerie Shaikly, and I'm very, very happy to have you here with us. How are you feeling tonight?
Yeah, good, thank you.
Thank you so much for agreeing and joining us tonight and bringing this topic, because definitely this is something that many patients wonder how it works when it comes to sperm donation selection. So Valerie here tonight will tell you a bit on that, of course, and she will start with her presentation. But as you know, afterwards, I believe it is your favorite part, because we will have a Q&A session. So as always, you will just need to type the questions in the chat section, and Valerie will help you with that right after the presentation. And let me just mention that Valerie is the founding partner of Fertility Genetics, and of course she will be able to tell you a little bit more on that as well. And remember, it's being recorded, so you will have a chance to watch this later on. It will be available tomorrow. And let me just add that my IVF Answers, as some of you might already know, is a part of European Fertility Society. There are over 300 webinars available on our site. And well, when it comes to European Fertility Society, we are simply here to support you, to educate you, give you as much information as we can, and of course give you this opportunity to ask your questions to those fertility experts, but also any experts that can help you out with many of those topics that we have. As you know, this week we are focusing on male fertility, and well, I guess sperm donation again will be another topic on that. And I guess now it is time to start with the presentation. Valerie, are you ready?
Yeah, I'm ready.
Thank you so much then, and let's go ahead.
Okay, so good evening, everybody. Thank you for the introduction, Caroline, and thank you very much to my IVF Answers for inviting me to speak this evening. I hope everybody will find the presentation useful, and I'm really looking forward to being able to answer your questions later. And hopefully, if I can, if I can't, I'll get back to anybody later. I'd just like to highlight that my background is UK based, but I think a lot of the information will translate to people whatever country you're situated in. I've been an embryologist for over 30 years, and I now practice as a consultant clinical scientist with a specialism in genomics, with the aim really of trying to make, perhaps difficult or tricky information, kind of present it in a way that's easier to understand so that people can make an informed choice for their fertility journey. I'd like to add that Fertility Genetics, founded by myself and my partner, is completely unaffiliated to any clinic or donor bank or genetic testing laboratory. Sorry, let's just move forwards. Okay, so now, yep, where do we start? So when you're looking for donor sperm, and actually the same often will apply to donor egg banks as well, actually, there's a very competitive market, big international market, and the information is kind of presented very differently according to the donor bank, and it can be very difficult to navigate your way around the websites to find the information in the first place, and then to perhaps compare and choose donors. So the aim of my kind of talk today is to kind of give an overview of the things that you should be able to see and what they mean, and how to look at them. So I think that every donor bank, basically the outline information that you would have would be physical and personal characteristics, and quite often these can be little tabs you have to click on. And then there will be tabs about the family health history of the donor. Then there'll be a section on genetic screening, and testing, and then lastly there'll be about the different options of the samples that you can buy, kind of different packages. So the first thing I'm going to talk about is the kind of physical, personal characteristics of the donor. That is probably the easiest thing to find and the easiest thing to navigate. So this generally will be perhaps as a summary, and it gives information about things like eye colour, hair colour, build, height. And then also on this tab, you will normally find information about blood groups for the donor, the ethnicity and heritage. So often this is just reported by the donor, but some of the donor banks do actually provide genetic testing to actually kind of validate, to say, well, actually we can see that the DNA of this donor is Northern European or Mediterranean descent. The donor, then they also have a pen portrait assay or essay. Basically, this gives the donor the opportunity to write about why they're donating, and a little bit about their kind of personal attributes that they wish to share. And this generally can be then obviously kept and shared in the future with children, if that's appropriate. The personality profiles are sometimes given. Sometimes this can be perhaps the impressions of the staff at the donor bank on meeting the donor. Sometimes the donor banks may actually provide psychometric testing that actually will kind of, for example, the donor may fill in a questionnaire, and from that they'll analyse that and perhaps give traits about the donor's personality. But these are all things quite easy to navigate and read through. The other piece of information that it can be key to look at is the family medical history of the donor, and sometimes this is just called a health history. So whenever a donor is recruited to a donor bank, they will sit down with a clinician or a genetic counsellor, and they will actually kind of elicit from the donor a two-generational family history. So this kind of goes back to grandparents of the donor, their aunts and uncles, cousins, parents, and their own siblings. And there's just a couple of examples here. So generally it would be a list of conditions kind of as a big list down one side, and then across the top it will list the relatives, and it's kind of a tick box exercise to say, you know, is this condition seen in the family, and if so, what member of the family? Some donor banks give a little bit more information than others. So some may actually, you know, perhaps describe the illness, how it was treated, if it was resolved, if it ended, you know, in the relative dying. So, you know, that there are different levels of information there. But what I would say is that at this point, if there's a significant family history of perhaps, I don't know, cancer across 3 generations or something that is significant, then a donor would not be recruited. So generally this is for background information. Sometimes though you may wish to consider things, for example, I don't know, for example, if there was a strong family history in your family of asthma and you saw that 3 or 4 members of the donor's family had asthma that were quite closely related to the donor, you may think, well, actually, we don't know what causes asthma. There's so many different things, but actually, I can see that, you know, perhaps I might choose not to have this donor. But on the other hand, you may feel that actually that's not important to you, and you'd be quite happy to proceed. And this is where I wanted to spend a little bit more time, is about the genetic testing and screening of the donor. So there's viral screening of the donor, which includes, which basically is looking at kind of sexually transmitted diseases. So no donors would be recruited if they had any positive antibodies for HIV, hepatitis, if there was evidence of syphilis, gonorrhea. So basically, donors would be excluded. So here we're looking at things that are kind of beyond that. So all donors will be tested for karyotype. So a karyotype is a test that actually looks at the number of chromosomes a person has, and is making sure that they have the correct number, and that they're structurally arranged in the correct way, so that, you know, bits of chromosomes haven't swapped places or perhaps inverted. And a donor would not be recruited if the karyotype was not normal. Just for those who don't know, so the chromosomes are the structures in our cells that carry all the genes, okay? And we have 2 copies of every gene, and we have 2 chromosomes of each number, 1 to 23. The other type of genetic testing that the donor banks can often report and provide is carrier screening. And this is for recessive conditions. And the number of conditions used can vary, but I'm going to explain that in a little bit more detail, or quite a bit more detail later. So I'll move on now to the things that probably is equally as important, is what the donor banks don't test for. So I have had people say to me previously, oh, well, I've chosen a donor and it's genetically screened, and so there shouldn't be anything. You know, I know there's no genetic disease, but that's not quite the case, because no testing will cover everything. The disease risks genes, for example, many people will have heard of the BRCA gene, or BRCA1 and 2. And if you have a change in that gene, that may predispose you to be at a higher risk of having cancer, breast cancer or ovarian cancer. It doesn't mean that you will get it, just means that there's a higher chance. So that isn't screened for, but hopefully that may pop up in a family history that was taken from the donor. Dominantly inherited diseases, so these will be diseases that if a parent had them, there's a 50% chance they'll pass them down to a child. And some of these can be adult onset, such as Huntington's disease. These aren't screened for either, but hopefully you would imagine that that would be picked up in a family history. But I think it's important to know that, you know, the family history is only as good as the donor provides, although they sign forms to say that they are providing information to the best of their knowledge. They may not have a huge knowledge of their family history. So I just think it can be, if you see on the health history everything ticked and nothing in the health history of the family, you could perhaps look at your own family and think, well, actually, if I was doing this, you would see a lot of things, and that's actually normal to have that. The other thing that isn't tested is multi-factorial diseases, so or conditions. So like I said, asthma, there's lots of causes of that. It can run in families, but it can be caused by lots of different genes or combinations of genes. And then, you know, with our environmental factors having an impact on top of that as well. So those things aren't tested for. So moving on to the extended carrier screening that I said I would come back to, is really to explain, firstly, I think, what a recessive disease is. So a recessive disease that lots of people are familiar with or have heard of is cystic fibrosis, and very common in the European, more common in the European population. Other examples of recessive disease would be sickle cell, anemia, thalassemia, etc. So to understand how a recessive disease works, I'm going to make up a disease and refer you to this diagram that we have there. And we're going to call the disease, we're going to call it, or the condition, we're going to call it purple. Okay, so you can see on the, oh, sorry, my apologies. You can see on the diagram here that both the egg and the sperm provider have the purple. They have one copy of the change gene, but they're healthy because your body can function just with one copy of the genes. So they also have a green gene, that's fine. So they're perfectly healthy. But if when eggs and sperm are made, only one of the genes will go into the eggs and sperm and then they combine. So in theory, if the two purples went to one child, we would have a child that didn't have a working copy of the gene. So they would be affected with the condition purple. You can see here that actually 50% of children would also be carriers of purple, and then we'd have one child in 4 or 25% that actually were unaffected but also are not carriers. So that's basically how recessive disease works, and this is why, because only 1 in 4 children are affected, you don't see it passing through a family line, because people can have children, 1, 2, 3 or 4 children. It's down to chance whether a child is affected or not. So you don't always see it in the family history, and it can be why these conditions kind of pop up out of the blue. I think when you look at the donor bank websites and you see a donor is a carrier, I think most people would be a carrier of something. It just depends how many genes you look at. So some donor banks, if you only look at 4 genes, you're less likely to find a carrier status than if you look at 283 genes. If we looked at 500, probably most people would be a carrier of something or other. So I think it's important not to be put off by that when looking at the donor status, but just to have an understanding about what it means. And what are your options really moving forward? So you found a donor, you've seen a positive carrier status. What do you do with that information? So my advice would be that if you do see a positive carrier status, is to find a professional that can kind of help you, support you around that information. And you could arrange a session with someone that would be able to assess, firstly, your personal medical history, take a family history from yourself, and look at that in the context of the family history of the donor. You could also review the testing results of the donor. So when was the testing done? Was it a long time ago? Which company was it by? How many genes were included? Around that information, and explain it to you. And also the limitations of testing, the accuracy of the testing at that time. Even not testing, even though they may be telling you that the donor is not a carrier of anything else, no testing is 100%. So there's always a residual risk that a negative finding can be positive and a positive finding can be negative, and it's about looking at those residual risks and understanding them. And then ideally what you would want that professional to do is to be able to say, well, we know this, so what does that risk mean to me? And if we go back to our example of purple disease, really the information that would help you make in your decision making would be to say, well, we know the donor is a carrier of purple. What are my chances of being a carrier of purple? And if I was a carrier of purple, what would be my chance of having an affected child? So if a condition had a carrier rate of around 1 in 50 people were walking around with the same, walking around with a change in the purple gene, there'd be about a 0.5% chance that if both the sperm and egg provider did have that a child would be affected. So here we're looking at low levels of risk, but they're known risks, and they're risks that can be reduced. So once you know that, some people may say, oh, I'll accept that risk, that's fine. I'm happy to use that donor. I perhaps might not have that information if I wasn't using a donor, or I could go to a donor bank where that information wasn't available. And they'll take that risk knowing that there's a 50% chance the child will be a carrier, and feel that that information may be valuable to them in their lifetime or for their child to know. Some people prefer to say, well, actually, I want to know if I'm a carrier of the same condition as the donor. And there's different options around that. You could say, ask just to be tested for the single condition that the donor is a carrier for. And sometimes people actually go about it completely the other way around. They may request that they do the kind of carrier screening initially. So they may, for example, a lot of the US donor banks at the moment screen for 283 conditions. So you could have the same test as the donor, find out what you're a carrier of, and then go and look for your donor based on knowing what you're a carrier of already. So there's different ways to approach it. If you are going to proceed to have the genetic testing to see if you are a carrier of the same condition of the donor or other conditions, again, it's important that you don't just order a test from a company from the internet and do it, because these tests need to be properly validated. The screening needs to match that of the donor bank. You need to they have to be ordered. I mean, tests that are kind of validated have to be ordered by doctors or clinical scientists. And you need to have that support if you do find a carrier status, because if you are found out to be a carrier of something, there's a likelihood that perhaps your siblings are, and what does that mean for your family? And the person who provides the testing should be able to sort of support you with that as well. So lastly is to look at the sample options that the donor banks will offer. So by this time, you've probably taken quite a long while to look at the different donor bank websites. You've chosen a donor, and then you think, right, now I'm going to order, I'm going to purchase. How much do I purchase? And oh my goodness, what do all these things mean? So again, as I said at the beginning, the donor banks are commercial businesses. So they tend to sell you the sperm by the number of motile sperm. So the higher the number of motile sperm, the higher the price of the portion or vial, straw of sperm that you purchase. And you may see the term MOT, and that basically stands for the number of motile sperm per milliliter. And I think another thing, you know, when you do actually buy a sample, is that you don't actually get a milliliter, you get half a milliliter. So if something says MOP 50, that doesn't mean you're buying 50 million sperm in your sample. You'd only be getting 25 million sperm in the sample that you bought. But why is this important anyway? Basically, it does depend on your treatment. So for some treatments, such as insemination, it's important that you have a higher number of motile sperm. Whereas for some assisted reproductive treatments where the kind of sperm and egg are placed together or the sperm is injected in the egg, so for IVF and ICSI, you need less motile sperm. And consequently, you know, those samples can be cheaper. But I would suggest that you would check with your clinic which kind of sample they would prefer you to buy. Another option around the samples is whether they are washed or unwashed. And all assisted reproductive clinics, when they're preparing sperm for use in treatment, will wash the sperm. So this means putting the sperm on a density gradient to try and kind of gather all the motile sperm together, and then they're washed, and then they're concentrated, so you have a high number of motile sperm in a small volume. And so again, it will depend on your clinic whether you purchase washed or unwashed, what they would advise, but generally most clinics will wash the sperm anyway before treatment. And then there's the number to purchase. As a golden rule, it's probably a good kind of guide is to say that one vial or a straw. So basically a vial is like a little tube with a lid that screws on top, and a straw is literally a small straw that's sealed at either end that contains the frozen samples. So one of those kind of portions is sufficient for one treatment cycle. But if you perhaps have purchased a low MOT sample and you were having IUI, a clinic may request that you have 2 per treatment cycle. So again, it's something you can just check with your clinic, but hopefully this just gives you an idea of what that means when you get to the point of purchase. So I'd just like to thank everybody for listening, and hopefully I will be able to answer your questions today. But if any questions pop up as you're going through looking at different donors, or you think about something, then please do contact me either through this format or through the My IVF Answers, or please feel free to contact me at info@fertilitygenetics.co.uk. Thank you very much.
So much, Valerie, for that presentation and for mentioning how exactly that works and how we should look at, yes, when we are selecting a donor. This is definitely very interesting. Thanks for bringing that. And well, there is time for the questions actually right now. So there are some questions ready, some interesting ones already as well. But of course, if you have anything, as Valerie said, you can ask this right now, but of course, you can also get in touch with Valerie, and I'm sure she'll be happy to get back to you as well. But yeah, let's not waste many more time, and let's go ahead with the very first and very interesting question. Angela, it's good to have you back. Always, you bring many nice and interesting questions. Okay, so if egg donor intended father don't have any common carrier disease, both did the Invitae 301 comprehensive carrier disease, including the add-on genes, would the surrogate, if they had any of those carrier disease of egg donor or sperm, be able to switch on one of the genes in the pregnancy? No one has the BRCA1 or 2. Is this also called ataxia telangiectasia, I think? Thank you.
Okay, so lots of questions there all in once.
Yes.
So I'll answer them as best I can as I go through. So if you have an egg donor who's been screened with the Invitae panel of 301 diseases, and the biological father or the sperm provider again has been screened, then the genetics kind of background of the surrogate won't make any difference. Okay, so they won't be able to switch anything on generally. There is a little bit of an argument about, obviously, there's the uterine environment. Does that affect perhaps the development of a baby? So that perhaps, you know, that the genes, does that have any impact on the kind of the genetic functioning of a child? And just so little is known about that. But for the diseases that are carried and carrier screening, definitely no. But there may be subtle things that we don't understand at the moment, that perhaps, you know, the environment that a baby grows in may affect which genes can be, as you said, switched off or on, or perhaps more active. But for the recessive diseases, the answer is no. So the BRCA, when you talk about BRCA, I'm not quite sure. So it's BRCA. So I'm 1 and 2. So I'm assuming that that's what the question is. And this increases your risk of ovarian or breast cancer. And I have to confess that I'm not quite sure what the ataxia telangiectasia is. But perhaps you may be able to get back to me, Angela, before the end and explain a little bit more, so I can perhaps give a better answer. If not, I'll get back to you after the presentation.
All right, thank you so much. Angela has added BRCA, of course. But of course, Angela, if you have anything else, we will go back to this one as well. Okay, so just let us know. And in the meantime, there is another question from Angela, so I guess we can go. Oh yeah, sorry, Angela has added something already. So I was told not to use any donor with ataxia telangiectasia.
Okay, I'll have to check that and get back to you around why you were told that. But I would have thought that if a donor has been recruited, and kind of gone through the recruitment process, that there wouldn't be anything, you know, unless you were a carrier of the same condition.
Okay,
that would be the only reason that they would tell you not to use that donor.
Yes, sorry, Angela has also added, as that gives an implication of future cancer.
Okay, so there are some recessive diseases that actually can slightly increase your risk of cancer, but there's a lot of other unknown factors around that, as in environmental, and perhaps other genes that kind of working alongside or confounding genes as well. So it may be that perhaps people that are a carrier of that condition, but I would need to check that. Something I don't know off the top of my head, I'm sorry.
All right, of course. Thank you so much indeed for, as I mentioned, interesting question. Yeah, extra cautious. Okay, thank you so much, of course. And well, let's have a look. Okay, another question we have from Angela. So, do you know why TISA is not accepted into guarantee live birth packages?
So yeah, a little bit off topic, but as an embryologist, I can answer that. So generally, Angela, I'm assuming that you're perhaps talking about live birth packages for the number of treatments and perhaps using donor eggs. The reason is because generally TISA sperm tends to be of a lower quality. So, you know, you don't actually see as many moving sperm. And, you know, perhaps the morphology of the sperm is not as good. So a lot of the live birth packages, so they can offer that, they will make the caveat to say that the sperm must meet WHO parameters or certain parameters, because they don't want sperm factor kind of there. They're offering you eggs and they're guaranteeing the eggs, but they can't then guarantee the eggs if the sperm doesn't kind of reach certain criteria.
All right, wonderful. Thank you so much once again for another question and your help with that. And of course, there's a thank you from Angela for you right here. Okay, and Bubble has another question. So do you know if the genetic screening of sperm donors is the same across European countries?
Okay, yeah, so I would say that in the European countries, no, it's variable. I'll count UK as being part of Europe as it was before December. But yeah, so for a lot of the UK banks, they will test for a minimum. They will only test for cystic fibrosis. I know some European donor banks will do carrier screening of about 48 genes. Some will do less than that, but some will also offer a carrier match, perhaps a carrier match option where you would go to them and you could pay extra to be tested to see if your carrier matched with the donor if you wanted to, but then you're not given the results of that finding. So you wouldn't know, but they can just tell you whether it's kind of safe to use or not. And I think that's because the laws in that country, I think specifically Denmark, they don't allow, if the donor's tested, then they're not allowed to find out their results. So if they did tell you your results, then the donor would have, you know, there's options there for that information to be found out. So yeah, that's another way of doing it. And I think, you know, you really need to look at the donor bank and kind of bury down into where, go under genetic screening, look at how many conditions there are. So I know the donor bank at the moment that screens for 47 conditions in Europe, 47, I think 46, 47. They will not, if a donor is a carrier, they exclude them. But when you're and I think there's a conversation around, there's a paper just came out on human reproduction last week, saying that, you know, this isn't sustainable because if we're going to carrier screen, we won't have enough donors. And actually being a carrier doesn't have an effect to health. So, you know, there won't be sufficient donors. So this is why the US banks who screen for more conditions actually give that information so that it can be kind of evaluated, risk assessed, and people have the information to make informed decisions about how they want to proceed.
And thank you once more for an interesting question indeed, and of course for your explanation to that. Definitely interesting. And next question is, do you know where I can find African sperm donors? I would like to get a non-anonymous donor, not sure where to look for.
Okay. So I think if you went to the kind of, I know that there is a shortage of African donors. I was actually speaking to a lady today who was you know, finding it difficult, but there are some, and so perhaps if you went to some of the big US donor banks, and if you're looking for non-anonymous, then if you kind of there's boxes to do your search criteria. So if you do your search criteria by countries that only allow non-anonymous donors, for example the UK, then they will come up.
All right, thank you for that advice here as well. Babel has added one more question. Let's go to that one. So what is your opinion on carrier screening of 600 genes for donor matching as offered by example iGenomics? Would you recommend it?
Okay, so the 600 genes, could I believe, I think, if just to check, is this where the donor has already been tested for 600 genes? Or is it something that you're thinking? So if you were, not many donors I know are tested for 600 genes at the moment. So if you were tested for 600 genes and the donor was only tested for 283, there's a chance that you could come up as being a carrier for something that the donor isn't, and then you're almost back to square one. But again, that information can be valuable. So thank you, Bubble. Yeah, so basically the egg donor and the husband can be screened. Yeah, so yeah, there is value. You're more likely to come up with a carrier status of an egg donor and a husband if you screen for more things. So, but the chances of them being matching, so going back to our example of purple, yeah, both being purple, for example, you know, that the more you look, perhaps slightly more likely to find that. But I would say that it's how far we want to look. So do we say that looking at 40 conditions is sufficient? Do we say 283? Or do we look at the whole genome? It's really about minimizing the risk that we can rather than eliminating it. So generally, donor banks at the moment are offering, as I said, some will only do one condition, some are doing 283, some are doing 40. So, you know, 600 probably is a top option at the moment. And if it's offered as a package, I think they call it like a co-carrier match. I think that's the one you're talking about. They basically look at it and then tell you, well, we've screened these 600 genes and you don't have alterations in the same gene that pose a risk to a child, and that's what they would tell you. So it would be the most risk reducing, but then you have to look at it from the point of view of expense and value as well.
And wonderful. Thank you so much indeed for that, again, advice here and your explanation. And someone is typing, so we just need to wait a bit for the next question, of course. And so if you haven't asked yet your question,
I think there's I know in Europe there's some clinics that actually are offering their own carrier screening, so they'll do it in-house, for example, and they say, oh, we're testing 1,000 genes, but then sometimes what can happen is you can find a change in a gene that people were not really sure what that means, and there's a question then mark over that. Do we know this causes disease? Is it likely pathogenic? That when it's called disease causing, it's called pathogenic. So is it pathogenic or likely pathogenic? And there may not just be that information. So you end up with lots of question marks and perhaps worry about something that might not happen or might not be important. And they're called variants of unknown significance. And when they start to get reported, when you start to get to really nitty gritty, then it can be a can of worms that can be really difficult to interpret and perhaps cause anxiety around things that perhaps wouldn't normally be considered.
All right, thank you for adding that. For sure, it's interesting to know. And in the meantime, as you can see, one more question is right here, and it's a bit of a longer one. That's why we just needed to wait a bit. And let's have a look. Okay, so I ordered straws from the European Sperm Bank out of Denmark and have done some screening from Invitae. Can't recall name. I was not carrier of anything notable. Donor screening was extra. Since straws have yet to be shipped, I can request the test, so I am responsible for making sure everything matches up, or the clinic, or do I supply my test result to the bank? Thanks.
Okay. I would contact, in this situation, I would contact the bank, because I think European Sperm Bank may be the one in Denmark, or be under the rules of Denmark, where they're not allowed to tell you what the donor is a carrier of, for example. Whereas if you submitted your test report on the official, if it's an Invitae report, with your correct name and date of birth, etc. on that, if you then send that to them, they will be able to check with their findings to see if there's a carrier match. And so that's the way I would do that, because the donor bank will perhaps have more information and understand how to do that perhaps a little bit better than the clinic, and you would have that reassurance before you purchase the donor as well then.
All right, again, thank you so much for yet
I think it does depend if the donors had the extended screening or not, actually. So, I think some of the donors will have had the extended carrier screening and some of them don't, and they call them, I can't remember, there's a special term they give on their website for a donor that's had the extended carrier screening. So first of all, check that. If they are offering the donor as having extended carrier screening, then share your report with the donor bank to say, can you check I'm not a carrier match. Okay, but I think they may not have used the Invitae panel, so it could be that that you've been tested for some things that the donor hasn't been tested for, and vice versa. And they would perhaps need to be able to look at that, and that their genetics team would be able to help you, I'm sure.
All right. Thank you once again for that, of course, thorough explanation once more. Okay, let's have a look. Okay, Kate has another question for you as well. So, yeah, we do receive that question. I remember actually it's definitely interesting. Can the same donor provide sperm for more families in the UK and outside of the UK?
Yeah, so basically the 10 family limit in the UK is limited to the UK, and then the donor will have its own kind of, every country may have its own regulations about how many families each donor can, but it can be cumulative across countries. So 10 in the UK, I don't know how many in the US are allowed, how many in Europe are allowed. So yes is the answer.
Okay, thank you for the clarification. So it is possible that someone can do it in the UK, but also you can actually travel abroad and also be a sperm donor, right?
So if you for example, if you're purchasing the 10 you have to purchase a pregnancy slot in the UK for a 10 family limit as part of the 10 families. Then, you know, if you do achieve your family in that, once you've got that, you hold that slot, that's yours, and you can use the donor for sibling use. But it does mean that the donor will be used to kind of for family building outside of the UK as well. And when you're perhaps looking about whether, you know, how many genetically related individuals there will be from that donor, and whereabouts they are in the world, that's something that it's very difficult to ascertain at the moment.
Exactly. And actually, next question is on that. Is there a maximum worldwide number for donors?
No, I don't think I don't even know whether it would be monitored worldwide at the moment. But I would think, you know, that in each European country, they would have their regulation for their own country, and then they may ship to the UK where there's a regulation. And, you know, the USA have their own donor banks. Unlikely that people might ship from the USA, from Europe. I don't know. Some of the European donor banks actually have outlets in the US where they, you know, you can order a donor from the US that can be shipped to Europe and then to somewhere else. So I don't think that is monitored at the moment. Definitely not worldwide. It may be within individual donor banks. They may have some policy around that, but I'm not sure that it's, you know, it is a requirement.
Right. Thank you so much. At least, well, this is not possible maybe in the future, because definitely, I mean, it's obvious that someone would want to know, yeah, if how it exactly works. But at least at this point, we just need to wait, and hopefully we don't know really. All right. I'm just looking if we have any other questions. Of course, we definitely want to make sure that you ask all of the questions. If not, we will be finishing for tonight. But of course, even if you don't have any questions now, remember that you can just simply get in touch with Valerie. You can use the link that I will put it in a second in the chat section, and I'm sure she'll be more than happy to help you out. And I don't see any more questions. I guess we will be finishing. And as you can see, there are just some things here from Bubble. Thanks very much for your explanation. Very helpful. That's true indeed. And Valerie, before we finish, anything else you can add?
I was just thinking a little bit more about the last question about limiting donor numbers, or perhaps, you know, finding out about that. And it's a little bit off topic, but is about direct-to-consumer genetic testing for ancestry now does mean that kind of no donor is really anonymous anymore. And, you know, if people are going to do something like 23andMe or some kind of DNA ancestry testing, there's a box you can click when you do that allows you to be visible to other people that share a percentage of your DNA. So cousins, you know, second cousins, third cousins. So if you perhaps, you know, you click the box and, you know, and you were donor conceived, you may find out that you got a percentage match with somebody who you didn't know you were related to. And through the kind of chats, it may not be that direct relative. It might be somebody else through the Ancestry website where you've ticked a box to be contacted. And then kind of with Facebook and social media, it doesn't take long to actually kind of work out where the donor is, or perhaps another donor conceived individual. So I think the anonymous and the non-anonymous criteria might be something a little bit of the past. From my own experiences, I did do 23andMe. I didn't tick the contact box, but if ever I log back in, it tells me how many more people are related to me. And I think when I did it 2 years ago, I last logged in, there was 600 people that shared my DNA, or elements of my DNA. So, you know, it's quite a powerful tool, and perhaps something that, you know, something to think about anyway, I think, when you're looking at the kind of anonymity side of the donation.
Yes, thank you so much for mentioning that. This is definitely a world that we're living in right now. So this is something we've discussed before as well. We've mentioned this before. We had some webinars where it was mentioned that nowadays it's really kind of easy to to find that thanks to those websites, yeah, like 123andMe. So thank you so much, and I guess we will be finishing for tonight. Thank you everyone for joining us, for your questions. As always, you've been wonderful with those questions, that's for sure. And of course, Valerie, thank you so much for a very interesting presentation with lots of details. And what can I say, it's been a pleasure to have you here for sure.
Thank you.
So thanks so much. And of course, one more thank you from Angela, but also from other patients for you. Thank you so much.
Thank you.
And well, as you know, it has been recorded, so of course you will have a chance to watch this again tomorrow. It will be available on our website, My IVF Answers, and if you subscribe to our YouTube channel, you will be notified that the video is uploaded. And of course, as you know, we will be back here tomorrow at 8 p.m. UK time. We do have another topic on male fertility, so I hope you will be able to join us as well and find out some more. Valerie, thank you so much indeed. Everyone, have a lovely evening, and I do hope to see you tomorrow. Thanks so much. Take care.
Bye.
Bye.